Okay, so in this video, we're going to look at protein synthesis and basically an overview of both transcription as well as translation. So when we talk about protein synthesis, what we're really talking about is how do we take the directions or like the recipe or the instructions of how to build proteins and actually make it happen in a cell. So protein synthesis is where we're going to start with the first step of taking that code from our DNA. Like I saw it once referenced in another video that a gene in our DNA is basically a recipe for making proteins. And I loved that analogy. And so here we're going to take that gene or the directions in our DNA and copy it into messenger RNA. And then you may have learned by this point in your biology career or experience that ribosomes make proteins and proteins are made of amino acids. So this process of transcription and translation is basically the how. So the two steps we have are transcription that we'll study first and then translation. So like I just mentioned, our DNA holds the instructions for how to make us. But our DNA is just that. It's only the blueprints or the directions or the recipe. In reality, like what makes our phenotype or our physical expression of our DNA, like what gives us our appearance and our traits, et cetera, are really the proteins that our cells make. The proteins are also what helps our cells to specialize. So proteins are super important in our bodies. And basically the DNA is just the directions of how to make them. So in our two steps, we're going to look at how do you start with a gene in DNA, which is our directions for how to make a protein. And basically, because our DNA is so important to keep intact, we don't use our DNA to make proteins directly, but rather we're going to make a copy of it so our DNA stays safe, and we copy that DNA into what's called RNA. So we're going to use messenger RNA. And then that messenger RNA is just that. It's a messenger, and it's going to carry those directions of how to build the protein to the ribosome because ribosomes are what build proteins. So the first step in going from DNA to RNA is called transcription. And then how do we read the messenger RNA to build a protein is called translation. So in eukaryotic cells, transcription happens in the nucleus where we find our DNA, and then translation where the ribosome, that looks kind of confusing, but where the ribosome will actually read the RNA, translation will happen in the cytoplasm. So if we look at the two kinds of cells on Earth, we have prokaryotes and we have eukaryotes. And all cells on Earth do transcription and translation. However, if you look at this diagram, you notice it's done in different locations. In bacteria or prokaryotes, they don't have a nucleus. So their transcription and translation will occur simultaneously in the cytoplasm. Versus in eukaryotic cells, transcription will happen first in the nucleus, followed by translation in the cytoplasm. So let's go ahead, though, and start with transcription. So we're going to start with our DNA, which is our gene or our directions for how to build a protein. And this is occurring in our nucleus in eukaryotes. So we're going to take RNA polymerase. That's the enzyme that is going to actually open up the DNA where the gene is. So we can use one side of the DNA to make the messenger RNA. So only one side will be used, and we call that one side the template strand. So really, if you're in a more advanced biology class, the RNA is made from 5' to 3'. So the DNA side we use will be the DNA side that goes from 3 to 5. If you're in a general biology class, and that's probably not too important to you right now. But basically, the RNA polymerase enzyme is going to open up the DNA. So it's going to break the hydrogen bonds between base pairs in DNA. And it's going to use only one side. Because if you remember from learning the differences between RNA and DNA, RNA is single-stranded. So here we use one side of the DNA, just one strand, to build the RNA strand. This right here is transcription, using the DNA to then make a copy into RNA. Now you notice in RNA, there are no T's. There are no thymine nitrogen bases. So on this last addition, watch as the, right here, how the A is going to pair with the U. That tells you that you're building RNA because there's only U's in RNA and there's T's in DNA. So then here you get to a T though, and it's okay. Oh, I'm sorry. So the RNA is actually going to start to trail off. Actually, if we go back, you can notice how like RNA polymerase is going to move and the DNA will wind back up and then the RNA strand kind of hangs off. But RNA polymerase will continue to build that RNA single strand. So now here on that T in DNA, there's an A that will base pair in the RNA. So this will continue until the end of the gene is reached. And then RNA polymerase will finish and the DNA winds back up into a beautiful double helix, safe and sound in the nucleus of eukaryotes. And now you have a single strand of RNA that is free from the DNA. So now that is it for transcription. Unless you're in a higher biology class, you might want to look at my video that's more specific on transcription that talks about RNA processing with introns and exons and stuff. Okay, so now though, this messenger RNA is made and it can actually leave the nucleus where it's going to then find a ribosome to do translation. So what we just saw is transcription in both prokaryotes and eukaryotes. Now, the next step is called translation. And in translation, we're going to translate that code into a protein. So here, if you look at the eukaryote, that messenger RNA is going to leave the nucleus and find a ribosome in the cytoplasm. So our next process we're going to look at is translation. And this is where ribosomes come into play. We learned that ribosomes make proteins. Well, how? It's not a random process. Instead, they're going to read the messenger RNA to build the protein with the right amino acid sequence. So here, ribosomes have two parts, a large and a small subunits. And they're going to like read the mRNA in codons. Okay, so a codon is made out of 3 bases. So here, our first start codon is AUG. And you can see that is 3 nucleotides. And these 3 nucleotides code for 1 amino acid. And in this case, it's methionine. Then the next codon we see is ACA. So a codon is made out of 3 nucleotides, 3 bases. ACA is the second one. And each codon codes for 1 amino acid. Okay, so basically the ribosome is going to read the messenger RNA codon by codon by codon to know which amino acids to bring. So there's actually a table. This table is called the genetic code. And the genetic code's job basically is to tell us each codon, like what each codon codes for the amino acids. So I'm going to write on this table for us. So like here, if I find like this codon, UUC, that is the group of 3 bases, and the amino acid it codes for is phenylalanine. So all of these like 3-letter abbreviations here, these are your amino acids. So serine, proline, methionine, valine, alanine, etc. So when you're using this table, this side here, these 3 letters, these represent the nitrogen bases in the RNA. I know it's RNA because there's U's, right? So you're never going to use the DNA for this table. And so this is how you're going to find your codon to see which amino acid it codes for. So for example, UCU codes for the amino acid serine. So let's go ahead and see 2 practice problems or practice codons to see which amino acid it codes for. So our first codon that we saw like here, our first codon was AUG. So let's go ahead and see, how do we know that that codes for the amino acid methionine? So here we have our table, and our first letter is an A, our first base. So there's the A. That means the amino acid we are looking for is somewhere in this row. Then our second letter, our second base is a U, uracil. So that means our answer, our amino acid, is going to be somewhere where these two, like this row and column overlap. Then my third letter is a G. So now where I have A-U-G, there is my amino acid methionine. So let's try again another codon. So here we have the next codon, C-A-U. So my first base, my first nucleotide is a C. That means my amino acid will be somewhere here. Then my second one is A. So where they overlap, my amino acid I'm looking for will be somewhere in this box here. Then my third one is a U. So then I have here, CAU is my codon. Histidine is my amino acid. Okay, so that's how you use the genetic code. All right. So let's go ahead and see translation. So how does the ribosome get the amino acids like there, right? So sure, the ribosome is going to read the messenger RNA, codon by codon by codon, but we need to bring the amino acids to the ribosome. So this process is called translation. So in translation, the ribosome is going to read the messenger RNA in groups of 3 called codons to determine which amino acid is next. So the first codon is always AUG. So you notice up here, there are a few other nucleotides, but we're not going to worry about those. We always start with AUG. This area is actually called the untranslated region. It's not translated. So here, we know by looking at that table that AUG coded for methionine. And so as the ribosome moves down, the next codon, GUC, codes for the amino acid valine. And then the next codon, AGC, codes for serine. And then CAC codes for histidine. And then eventually, though, you get to a codon that says stop. Hey, the protein is done. Because if we remember, proteins are made of amino acids. So here, this messenger RNA was just the directions telling the ribosome the order to add the amino acids together. So here you have codon by codon by codon. Each codon is telling the ribosome which amino acid. Now, the average protein in eukaryotes, I think, is like 400 amino acids. So that would take like 1,200 nitrogen bases to get 400 amino acids, but it'd be like a longer chain. And so the size of the protein is really dependent on how long is that messenger RNA and how many codons are there. Now, these codons also tell the ribosome which amino acids to join. Okay, so now let's go ahead and practice a little bit. Oh, no. So our question now becomes, though, like, how do those amino acids get there? Like I just showed us, like if you use that little table, AUG codes for met, but how does methionine actually get to the ribosome? So here, there's another type of RNA called tRNA. And tRNA stands for transfer RNA. And transfer RNA's job is to bring the amino acids to the ribosome. So this blue line here represents the RNA. And I kind of color coded it because we know our ribose sugars here are blue. So this is like a single strand, just like this single strand, but it's folded up until like it actually kind of looks like a T shape. So this transfer RNA is one strand of RNA that's kind of folded up. Now RNA though is made out of nucleotides, right? Like this here, this is nucleotides, and that's what this blue line is made out of. So at the bottom of this tRNA, there are 3 important nucleotides. Now, the bottom of the tRNA, as the RNA folds up, we call this area the anticodon. So at the bottom of the RNA, the bottom 3 bases we call the anticodon. So let's just look for a second here. So like here you can see it's a single strand of RNA, and at the very, it kind of like hydrogen bonds with itself, like to hold it in that like T shape. But down here at the bottom, these 3 bases, in this picture it's GCC. This is the anticodon. And so basically what we see, or what we will see, is how that anticodon is going to pair with the codon. Now the tRNA, at the very top of the tRNA, that's where you find the amino acids. like it's the tRNAs bringing the amino acids to the ribosome. So here we go. Let's find our first start codon, AUG. Now, we can use that genetic code table, and what we'll find is AUG. This is kind of blurry, but it codes for MET. So MET is our start codon, and that's our first amino acid. Now, our next codon is GUC. So we would use the table, and we would find G, and then UC. Oh, that's valine. So you can tell here at the top of this tRNA, there's a valine there. That yellow circle is the amino acid. And I want you to pay attention, this yellow box here, here's the codon in the messenger RNA, the group of 3 bases. And look how the anticodon right here at the bottom of the tRNA matches to it. Now the ribosome, before it moves down, what's going to happen is between methionine and valine, they're going to form a bond. That's a peptide bond, actually. It's a covalent bond. Now, the tRNA that carried MET to the ribosome, MET right now, when this bond forms, MET or methionine is now joined to valine. We're forming our chain of amino acids. So that means that this tRNA is now empty. So the next time you have dinner or lunch or a snack that has protein, you'll get new methionine amino acids, and an enzyme will actually attach a new MET to the top of this tRNA. So here, though, the ribosome is going to move down the RNA, the messenger RNA, to the next codon. And this is where we're going to use that table, AGC, to find which amino acid is it. AGC, right here. So that's going to be serine at the top. So in this tRNA, we can see here that we have an anticodon, UCG, that's going to pair with our codon. We also see that eventually valine was going to form a peptide bond or a bond with serine. So valine is now joined to serine. We're forming our chain of amino acids, which means now that this tRNA is going to then be free and empty, basically. And then the ribosome is going to move down to the next codon. Okay, let's clean this off a little bit. It's going to move to the next codon, which is CAC. We'll use the table to figure out which amino acid is arriving next. CAC is histidine. So at the top of the next tRNA, we should expect, that one leaves, sorry. But we expect histidine. Oh, I had my animations out of order a little bit. That one moved a little too early, sorry. But eventually, we're going to reach a stop codon. Now, when the ribosome reaches a stop codon, that says, hey, stop. We're all done. We've reached the end of our directions. We're finished. So in this picture here, there's only 4 amino acids forming our protein or our polypeptide. So in reality, we have hundreds or even thousands of amino acids joined together into a chain of amino acids called a polypeptide that will fold up and form our protein. Now you can see how it was the codons. It was the order of the codons that, or the sequence that told us how to join, what order to put the amino acids. So the directions came from our DNA, and they were transcribed or copied into RNA. And now the ribosome read the RNA to know which sequence and how many amino acids to join in our polypeptide. So when we look next at the overview of this, we start with DNA in our nucleus, and it's a double helix. There's 2 strands. So I know even if I didn't see this word here, I would know this is DNA because of double strands and the T's. So in transcription, the DNA kind of opens up by that enzyme RNA polymerase, and then you have messenger RNA will be transcribed. I highlighted the U's here to help you point out the difference between DNA and RNA. So you can see how uracil is found in RNA and thymine is found in DNA. Now this step right here is transcription. That's what transcription is, to start with DNA and then make it a copy of RNA. Now the DNA though will go back to a double helix, and the RNA will go to a ribosome. So if it is in a prokaryote, a bacteria, the ribosome is probably already attached and started reading it. But if it's a eukaryote, the messenger RNA will be edited a little bit. That's a higher level biology topic, but it'll be like chopped up and reorganized a tiny bit. But then the messenger RNA is going to leave the nucleus and travel to a ribosome out on the rough yard or in the cytoplasm. And that ribosome is going to read it codon by codon by codon by codon, because each of these codons codes for an amino acid. So now we are actually using those directions in the DNA to figure out what is the sequence or the order of the amino acids coded for by this gene. So AUG codes for meth, then CGU codes for arginine, and then here we'll just use one, CAU codes for histidine. Then we have GCC codes for alanine, and then eventually we get to a stop codon, telling us, okay, stop, your polypeptide or your protein is done. So this second step here, when we went from messenger RNA to an amino acid sequence, this is called translation. All right, so these are our two steps involved in building a protein, and this whole process is called protein synthesis. So great job, my wonderful students.